Non-stimulatory peptide MHC complexes do not activate T cells but can enhance T cell responses to agonist peptide MHC. This protocol describes an experimental system to investigate co-agonism during human CD8 T cell activation. We expressed MHC molecules as single-chain complexes with covalently linked heavy chain, beta-2 microglobulin, and the peptide. This allows introducing mutations into MHC molecules presenting predetermined peptides.
This protocol uses human CTL clone specific for an epitope of hepatitis B virus. Understanding the mechanism of co-agonism during T-cell responses against hepatitis can contribute to development of immunotherapies against this viral infection. The presented methods can be used in research on other fundamental aspects of T-cell activation in human T-cell systems with known peptide MHC specificity.
This protocol uses single-chain human MHC class I molecules, consisting of signal sequence, peptide of interest, linker, human beta-2 microglobulin linker, and MHC heavy chain. E183 peptide from hepatitis B is used as agonist peptide. GAG from HIV is used as non-stimulatory peptide. The single-chain human MHC molecules are expressed in a hamster cell line containing tetracycline repressor. Agonist single-chain MHC is expressed using tetracycline-inducible plasmid. Result